Hello and welcome to this first online lesson during the school closure. This lesson is intended to run as a lesson normally would that you would have with myself, Mr. Abraham. So we're going to start by looking at our learning objectives. Up until this point we have been looking at how antibiotics, painkillers and vaccines work. We've spoken about how drug trials can help us to create new drugs, and today we need to look at monoclonal antibodies. Now, monoclonal antibodies is only found on a higher tier paper, therefore, because if you're in set 1 or if you're looking to do a higher tier paper for biology, monoclonal antibodies is a big part of that. So in your books, I would like you to write the title, please, Monoclonal Antibodies. I'd like you to put the date. I am intending to use an online chalkboard, so I will be using the same colour-coordinated system that I would use in any other lesson. So today's title, again, monoclonal antibodies. The learning objectives are, by the end of this lesson, you should be able to recall what a monoclonal antibody is. You should be able to explain how monoclonal antibodies are produced, and you should be able to tell me some strengths and weaknesses of using monoclonal antibodies. Now, we're going to start this lesson off with a keyword, so I'm going to put it in green. Our first keyword is the word monoclonal. Monoclonal antibody or antibodies. Now, before we break down what this is and before we define it, I just want to break down each individual word so that we have a better idea of what it means. The word mono means one or singular, okay? Clonal means a copy of something. So a copy. And antibodies are special proteins that attach to a specific antigen. So the final bit is special protein specific to an antigen. So what we've got is lots of copies of a single antibody. So we're making many copies of an antibody that is useful to us. So we can define monoclonal antibodies as antibodies produced from a lymphocyte or from a hybridoma, which we'll talk about in a moment, from a hybridoma cell, that are specific to a antigen. So monoclonal antibodies are antibodies produced from a hybridoma cell that are specific to an antigen. Now the point is that these monoclonal antibodies only fit a single antigen. You can't get monoclonal antibodies that fit multiple antigens. They only fit one. So although I've already used red here, I'm going to use it underneath, and this is supposed to encompass everything in our definition. We can say that monoclonal antibodies are specific to a specific antigen. So monoclonal antibodies are specific to a specific antigen. And what this means, So we can put comma. What this means is that they only bind to a single type of cell. They don't tend to join to different types of cell. We can target cells to bind these monoclonal antibodies to. So we can put, as a result, they bind to specific cells. They bind to specific cells. Okay? So monoclonal antibodies are antibodies produced from a hybridoma cell, which we'll come to in a moment, that are specific to a antigen. We've said that monoclonal antibodies are specific to a specific antigen, and as a result, they only bind to specific cells. We can dictate which type of cells we're looking for by the antibodies that we make. Now, monoclonal antibodies are used for three main purposes. So if I can just change my colour, we'll go for this nice turquoise. I must admit, the one benefit to using this style of teaching is I have so many more colours in this colour palette than I do my normal ball pens. I'm loving it. So we can put monoclonal antibodies are used for, so monoclonal antibodies are used for The first thing they're used for is diagnosis. So number one, diagnosis. Now, we use monoclonal antibodies for things such as pregnancy tests. Pregnancy tests look for a substance known as HCG. There is a video at the start of the PowerPoint attached. If I show you the PowerPoint, in this starter, if you click on this link up here, this will give you a video on how pregnancy tests work. But we can use monoclonal antibodies to look for a molecule known as HCG. So we can put next to diagnosis, we can put e.g. diagnosing pregnancy. And we can say that, put, for example, in brackets, a molecule known as HCG. You may wish to look into that a little bit more. Another use we have for monoclonal antibodies is we use them in research. So number two, we can put research. Okay. Now, we can use monoclonal antibodies to either locate or identify specific molecules within a cell. We can actually attach dyes to the monoclonal antibodies so that when they then attach to a specific molecule, they glow up. When they glow up, we can see where they are in your body. So if we're looking for a specific molecule within a cell, we can get the monoclonal antibodies attached to a dye. That monoclonal antibody will attach itself to that molecule. That molecule will then glow up and show up on any scans. So we can put, next to this, we can put locating and identifying specific molecules. So locating and identifying specific molecules by adding dye to the monoclonal antibody. The final use for monoclonal antibodies is in treatment. So number three is treatment. We can use monoclonal antibodies to provide treatment for illnesses. Okay, so number three, we can put treatment. Now, what we mean by treatment is we can add drugs or we can add radioactive substances, allowing monoclonal antibodies to treat cells that have cancer, for example. If we know there's a cancerous cell inside your body, we can attach drugs to the monoclonal antibodies that we'll use. They'll go away, attach themselves to the cell, treating it, either providing it with a drug or providing radiation. So we can put here, adding drugs slash radioactive substances, to monoclonal antibodies can treat things like cancer. So let's have a look back at our learning objectives and have a look at how far we've got through this lesson. So if we have a look back at this, you should now be able to tell me what a monoclonal antibody is and what we can use it for. So a monoclonal antibody is antibodies produced from a hybridoma cell that are specific to an antigen. Monoclonal antibodies are specific to specific antigen, and as a result, they only bind to specific cells. Therefore, we can use monoclonal antibodies for diagnosis, such as pregnancy, research, finding where certain molecules are within a cell, and treatment by adding drugs or radioactive substances to the monoclonal antibody and allowing it to join to things like cancer cells. The bit that we need to have a look at now that we haven't really spoken about yet are these hybridoma cells. So the hybridoma cells are something very, very specific, and in the questions that I've seen at GCSE Biology, you mostly need to be able to say what a hybridoma cell is and how it is produced. So if we can just refresh this screen, can I just point out, if at any point there's something you don't understand or you didn't quite get, remember you can pause this video. It's not like a normal lesson. You can pause the video and just say, oh, I didn't quite get what a monoclonal antibody is. Let me rewind it and have a listen again. So remember, you don't always have to just listen to this all the way through. Now, to help us with this, for a nice diagram here that I stole offline or out of a book, I can't really remember if I'm honest, and I'm going to stick this to the side while we talk about it because it'll come in use in a moment. So if I can just shrink it down a little bit and we can move it over here out the way, and we'll refer back to that later. Before we do anything, before we talk about these hybridoma cells, you need to understand that lymphocytes lose the ability to divide into new cells once they produce antibodies. So stem cells, for example, can divide into any type of cell, but once they divide, that's it. They can no longer divide into a different type of cell. Lymphocytes are the same. Once they find a antibody that fits a particular disease, they can no longer change what antibodies they make. So the first thing we can write down, and I'm going to put it in red because it's really, really important, we can say lymphocytes, so lymphocytes lose the ability to divide, into new cells once they produce antibodies. So if you think back to when we spoke about vaccines, we said that when you're initially exposed to a foreign antigen, your lymphocytes start to produce lots and lots of antibodies. Once they find an antibody that fits that antigen, they can no longer make any other type of antibody. So lymphocytes only tend to produce one type of antibody once they know how to make it. This is really important, and this is a really important concept you need to keep in mind. Okay? So our subheading for our second learning objective, we can say making monoclonal antibodies. I'm going to use a different colour so I can number it. Making monoclonal antibodies. Okay, making monoclonal antibodies. So the first thing that we do when we make monoclonal antibodies is that we inject a mouse, which is shown on this diagram here. We inject a mouse with a specific antigen so that their lymphocytes make antibodies that fit it. So number one, inject a mouse with antigens that we want antibodies for, full stop. And we can say that the mouse lymphocytes make antibodies that fit. Make antibodies that fit. But the problem is that these lymphocytes no longer make any other type of antibody. And at the same time, we've only got one lymphocyte that does it. We want to make lots of this type of antibody to fit the antigen we want. This might be an antigen for a cancer cell, this might be an antigen for HCG in a pregnancy test, it might be an antigen for a specific molecule found within a cytosol. So we want to make lots of these lymphocytes. We need lots of them. They're all making antibodies that fit. Okay? So we want lots of them. So want lots. Okay? Because one is not enough. We want to make millions, if not trillions, of these antibodies. So what scientists do is they take cancer cells, they take cells from a tumour, and they join these lymphocytes to a tumour cell. Now, tumour cells are very, very good in the sense that they keep going through mitosis. So if we join the tumour cell with a lymphocyte, we should get a lymphocyte that just keeps going through mitosis and it produces lots of antibodies. So we can say here, number two, extracted lymphocytes are joined to tumour cells. So we join these extracted lymphocytes to tumour cells. Now this forms what is known as a hybridoma cell. Can I point out this diagram is on the PowerPoint that I've sent to you, so if you want to see this bigger you can. So this makes what's known as a hybridoma cell, and that's our next keyword. Okay, hybridoma cell. Now a hybridoma cell is defined as a cell made by combining tumour cells with lymphocytes to produce a cell, that makes antibodies and, importantly, divides. So the important thing is that not only does this hybridoma cell now make the antibodies we want, but it can also divide. So we can then put for number 4, which is here, that the hybridoma cell divides. Hybridoma cell divides by mitosis, forming lots of cells that make antibodies, or that make monoclonal antibodies. That make monoclonal antibodies. Now these monoclonal antibodies are shown down here. Okay, so these are lots of monoclonal antibodies that are collected. So for the final stage here, number 5, we can say that the monoclonal antibodies are collected. So monoclonal antibodies collected. Now these monoclonal antibodies, sorry about that, that should say collected. These monoclonal antibodies can then be used for diagnosis, they can be used in research, they can be used in treatment. So to just run through this, lymphocytes lose the ability to divide into new cells once they produce antibodies. So we want lots of lymphocytes all producing the same antibody so we can use them for diagnosis, research and treatment. To do this, we inject a mouse with the antigen we want to search for. So the antigen might be from a cancer cell, it might be HCG inside of a woman's urine to detect pregnancy, it could be lots of things. But we inject that into a mouse. As a result, the mouse's lymphocytes make antibodies that fit the antigen. We want lots of these. We can then extract these lymphocytes and join them to tumour cells. Tumour cells go through mitosis very, very quickly. So we've got lymphocytes that can't go through mitosis but produce lots of antibodies, and we've got tumour cells that can't produce antibodies but do go through lots of mitosis. We join them together to form what's known as a hybridoma cell. A hybridoma cell is a cell made by combining tumour cells with lymphocytes to produce a cell that makes antibodies and divides. So we've now got a cell that not only produces the antibodies we want, but it also divides, which is exactly what it does. It divides into lots of clones of the original cell, which can all produce antibodies. Each of these start making antibodies, therefore we have lots of them, which we can go on to use. So if we have a look back at the learning objectives, it says, can you recall what a monoclonal antibody is? Can you explain how monoclonal antibodies are produced? The final bit is that we need to evaluate the strengths and weaknesses. So if we can just refresh this, remember you can pause this video, you can go back and re-watch parts if you didn't quite get it. If you're still stuck after watching this video, you can either comment in the Google Classrooms, or you can drop me an email and I'll reply as soon as I can. So let's have a think about some strengths and weaknesses of monoclonal antibodies. So we can divide our page up like this. On one side we can have our strengths, and on our other side we can have the weaknesses. For strengths, it's fairly straightforward. We've already spoken about them. We can use them to diagnose that we otherwise wouldn't be able to. If you watch that video on pregnancy or pregnancy test, you'll see how the Egyptians used to do it compared to how we do it now. We can use them for research. So a lot of what we know about cells is based off using monoclonal antibodies in research. And finally, we can use them in treatment. So lots of treatments rely on monoclonal antibody to help us do that, things like cancer. Now the sad side, or the not sad side, but the weaknesses of using monoclonal antibodies, they can have side effects. So the side effects of using monoclonal antibodies, so side effects, these can include nausea, so being sick. These can include rashes. They can include fever. They can include breathlessness. And they can also include things like headache. So headache. Now this is mainly because it's our body trying to reject the mouse proteins. So all these side effects are due to the body rejecting mouse proteins. Okay? So those are our strengths, those are our weaknesses. The strengths include that we can use them for diagnosis, research and treatment, whereas the weaknesses are that they cause the following side effects. So you should now be able to tell me what a monoclonal antibody is, you should be able to explain how monoclonal antibodies are produced, and you should be able to tell me some strengths and weaknesses of monoclonal antibodies. Please, if you are stuck with anything, drop me an email or reply in the Google Classrooms. I've attached some worksheets as well to Google Classrooms for you to have a go at. I may work through them, however I have got a lot of videos to make, so I may end up just putting the mark schemes in there. As always, you need to make sure you work through the worksheets and mark your own work. If you are stuck with anything, it is your responsibility to either contact me on Google Classrooms or drop me an email. In Google Classrooms, if you like these videos, if you like the way that this is being done, please let me know, because if you don't like it done this way, I'm open to trying anything else. This is new to all of us. This is ultimately, it's your education that matters. You guys have got your exams in 2021 in about a year's time. It's really important that you don't lose any of your education due to this unfortunate circumstance. So again, please let me know if you want me to change anything. Please let me know if you're stuck. I will put another lesson up when we are due to have one. For now, I would like you to have a go at the questions that I'm going to put on this PowerPoint, and I would like you to have a go at any worksheets that I attach in Google Classrooms. I will see you in the next lesson. Thank you.